Hello everyone, welcome back. We're talking about the spinal cord development and now we'll deal with histogenesis of the neural tube. Histogenesis actually includes the type of cells which actually help in formation of the neural tube. We'll see what are different types of cells, from where they originate and how they participate in the formation of neural tube. We'll see what are the current and the old theories of development of the spinal cord and how these theories help in understanding the histogenesis and the development. The spinal cord actually develops from the caudal elongated part of the neural tube. So the neural tube, when it's formed, it gives rise to a cranial enlarged part, which gives rise to the brain vesicles, and a caudal elongated part, which gives rise to the neural tube. This is the developmental figure which shows the alar plate, the central canal, the intermediate plate, the basal plate, and the marginal layer. This is a developing or spinal cord which finally develops into this structure. This is the central canal. Now this is the gray matter, this is the white matter, this is the lateral horn, this is the anterior horn, this is the anterior or the ventral column, this is the central canal. So the spinal cord develops into the structure shown in the figure. We'll see how this development takes place. So in histogenesis, there is repeated mitosis which leads to thickening of the epithelial lining. So this is the epithelial lining which undergoes repeated mitosis and it undergoes thickening. So three zones are formed. These are the matrix zone, the mantle zone, and the marginal zone. So thickening of the endoderm results in due to repeated mitosis results in formation of three zones. Let's take a look on these zones. Now the duration is middle of the 5th week. Now the transverse section of a closed neural tube reveals a matrix zone. This is the matrix zone. Then we have the mantle zone, this is the mantle zone, and finally we have the marginal zone. This is the marginal zone. So let's see what happens in which zone. So first of all, we have the matrix zone in yellow. It is thick and it lines the neurocele. Neurocele is the central opening which gives rise to the central canal in the adults. So neuroblasts develop to neuron in this area. So the neuroblasts develop to neuron and the spongioblasts develop to the neuroglial cells, the supporting cells of the central nervous system. Now the neuroblasts, the cell bodies of the neuroblasts, they migrate to the mantle zone. Here, this is the mantle zone where the cell bodies of the neuroblasts, they migrate. They are actually formed in the matrix zone and they migrate into the mantle zone. Here you can see this is the mantle zone, this dotted area. Now the axons of these neuroblasts, they enter the external marginal zone. This is the external marginal zone. Here you can see these are the axons entering the external marginal zone, which is the future white matter. So this is how gray and white matter are differentiated. Gray matter contains the cell bodies and is formed of the mantle zone, while white matter, it contains the axons and it is made up of the marginal zone. Now there are some central processes that ascend in the margin. Central process of the dorsal root ganglion that ascend in the marginal zone, and other synapse with the neuron in the mental zone. So there's various interconnections between the cell bodies and the axons and the various neurons which are present in various layers. These interconnections are important because they help in the carrying and relaying of information to and from the central nervous system. So talking about old and the current theories of development. Now once complete, the remaining matrix cells, they differentiate into ependymal cells which line the central canal. So once the formation of these zones is complete, the remaining cells now line this central canal and they form ependymal cells. So the current theory is Wall of the recently closed neural tube have only one types of cells, which are the pluripotent neuroepithelial cells. So this is the current theory, that a wall of the recently closed, newly closed neural tube has only one type of pluripotent neuroepithelial cells. They extend over the entire thickness of the wall and form a thick pseudostratified neuroepithelium. Now these one type of cells, they extend over the entirety of the thickness of wall and form a pseudostratified neuroepithelium. Now this zonal appearance, the zones we talked about, these matrix zone, the mantle zone, the marginal zone, it merely reflects different phases of the proliferative cycle, that is interkinetic migration. So according to this new theory, when this neural tube is formed, it consists of only one type of cells. These one type of cells are actually pluripotent. Pluripotent meaning they can give rise to a variety of cells. So these pluripotent cells first form the neuroblast and the neuroglia. These neuroblasts and the neuroglia then move to the mantle zone, and leave the cell bodies there, and then the axons enter the marginal zone. So technically this zonal appearance that is actually seen in the development of spinal cord is basically just the interkinetic migration of these cells. These cells migrating through different layers to come to lie in their adult positions. So this is the current theory of CNS development. Now, again summarizing all of it, the neuroepithelial cells, these neuroepithelial cells, they give rise to the neuroblasts. These neuroblasts, they surround the neuroepithelial layer to form the mantle zone. So the mantle zone has been formed. Now mantle zone forms the gray matter of the spinal cord. Now the marginal layer, it undergoes myelination of the nerve fiber, and the white matter of the spinal cord has been formed. So summarizing it, what happens is the cells, they start from the central zone. The neuroepithelial cells, they actually form the neuroblast and the neuroglia. The neuroblast and the neuroglia come to lie in the mantle zone, and and leave the cell bodies there. From there, the axons tend to move to the marginal zone. In the marginal zone, there's myelination of the axon fibers, and this is responsible for the white matter or white appearance, or the production of white matter. Since the mantle zone contains the cell bodies are non-myelinated, and this appears as gray area of the brain matter of the spinal cord. So this was all about this section. I hope you understood this section. For further such sections, keep watching sqadia.com.